Okay, so what we want to go through this time is how we actually classify our epithelial tissue. An epithelial tissue can be classified based on two different things. The first thing is the number of cell layers that are present. And so when we actually construct the name for our epithelial tissue, the first word in that name is actually going to indicate to us how many layers are present. And the second part has to deal with cell shape. So the second word in the name of any epithelial tissue will be the shape of the cell. So let's talk about the words that are used for these classifications. So when we look at layers, the first option is simple. And when we talk about something being simple, we talk about it being a single thin layer of cells. And they are only found in these protected internal areas, simply because the more deep they are within inside the body, the less chance they're going to have to be damaged by abrasion. So a single layer will provide some protection, but if it's in a harsh environment, like on the outside surface exposed to the outdoors, then that's actually not a good thing. The second characteristic is stratified. Stratified means that we have several layers, and these are actually much more of a protective function. So they can actually exist in areas that have a little bit more need for durability, like the skin, the surface of the skin, inside of our mouth, and around the anus. So let's take a look now at the cell shape. So those are our two options, either simple or stratified. And one of those words is always going to be the first word when we actually name something. The second word, we have three options, and that's based on the shape. The first shape is squamous, and squamous basically means that I have a thin, flat cell with the nucleus in the thickest part of the cell. So if I look at the cell from the side, I actually have kind of a fried egg look. So let me do a quick drawing here. So it would look something along the lines, actually here, so you guys can see it, like this, where we would have the cell and its borders, okay, and in the thickest part here would be that nucleus. Now, if I looked at it, let's say through a microscope, what I would see is I actually have some kind of jagged borders, okay, and things can be pretty much any shape. They can be like this in a triangle. They could be kind of a square shaped, but they're not going to be any one specific shape. They're also not nice, neatly arranged, which we will see with some of the other possibilities. The second possibility is cuboidal. And cuboidal means that we actually have cells that through the microscope look like squares. And if you think of these cells in 3D, then of course it's going to be a square in 3D as a cube. So that's where it gets its name from. These do exist in very nice, neat rows, with the nucleus, which is usually fairly large, very predominantly in the center. The third option is for a columnar configuration. And these are tall, slender rectangles. And the nucleus does tend to be near the bottom, down near the basement membrane, but it doesn't have to be. Sometimes you will actually find that it's kind of floating out in the middle. So based on these three shapes and the two types of layers that we've got, we actually can classify our epithelial tissue by creating the different combinations. So let's look first at our first combination, which is simple squamous. So I have one layer of squamous shaped cells. These function in absorption, and they also help to reduce friction. Because it's a simple layer, we do find these in protected internal areas because it's not very durable. This would be places like our kidney tubules, the lining of our ventral cavity, which is our front body cavity where like our heart and lungs are, as well as our digestive tract, the lining of the blood vessels, as well as the inner surfaces of our heart. And then down below that you have actually a picture of simple squamous, and you can see we've got some different shapes here of the cells. The borders are kind of ragged, and they're not all one consistent shape, and that's very typical of simple squamous epithelium. The second type would be simple cuboidal, and these function for secretion and absorption and some limited protection. They're not quite as thin because they're a cube as the squamous cells, so there is some protective function. We find these cells in the pancreas, the salivary glands, and the salivary ducts, certain portions of the kidney tubule, specifically the ones that are involved in urine production. And again, we have a picture here of the simple cuboidal epithelium. Okay, we've got the very nice, neat rows of square cells with a very large predominant nucleus. The third type of simple epithelium is simple columnar. And just because the rectangles do tend to be pretty thick, they do provide some protection, but mostly they're involved in absorption and secretion. These tend to line the stomach as well as within our intestinal tract and our excretory ducts. And you can see here, this one is actually a picture from the intestine, and that's why you have the microvilli there. It looks like kind of a dark pink line at the top, but you've got these tall rectangle-shaped cells that are all in a nice, neat line. All right, so we have 3 more to go in terms of our combinations, and that is the stratified. Stratified squamous is the first one, and these are areas where we have very severe mechanical stresses, like the skin, the lining of the mouth, the surface of the tongue, the lining of the esophagus, and around the anus. These tend to have outside foreign objects that actually brush up against it very often. Okay, and you can see we have multiple layers of cells. Now the cells down towards the bottom, towards the basement membrane, those stem cells do tend to look square in shape, but as you look up through the layers, remember they're not changing the type of cell that they are, and so you'll see they lose that kind of square appearance as they move up, and that is very typical of a squamous cell. So we also have stratified cuboidal, and these are very rare within our bodies. We find these, for instance, in sweat gland ducts and in lining some of the larger ducts of the mammary glands. So again, we would have nice, neat rows stacked on top of each other of nice square cells. Stratified columnar is also rare within the body. We find these in portions of the pharynx, which is like the back of your throat, the epiglottis, which is the flap that actually covers your trachea and prevents food from going down into your lungs, the anus, the urethra, and a very few of the large excretory ducts within the body. Now, if you take a look, we still have 2 more types of epithelia, but these are not classified based on their shape and the number of layers. These are actually 2 special cases that we want to look at. The first one is called Pseudostratified Epithelia. And if you think of the prefix pseudo, it means not really. So basically, it means it's not really stratified. So this is epithelial tissue that looks stratified, but when you actually really take a good look at it, it's not. It is typically a mixture of columnar cells that have cilia on them. And that actually creates the full name, which is Pseudostratified Ciliated Columnar Epithelium. It's kind of a mouthful, but it's really the only really weird oddball out there. We find these in the nasal cavity, the trachea, the bronchi, which are the tubes that branch off of the trachea. So a lot of the respiratory system has this, which is why it's important for us to know, as well as within the male reproductive tract. And you can see here with the picture, these are the only types of cells that we have that have cilia. And if you look down here at the bottom, it does appear that they would be stratified, but it's really not. The last type of epithelia, true epithelia, is transitional. And this is another special case. This is stratified that actually tolerates repeated stretching. So the appearance of this actually changes, in other words, transitions, which is where it gets its name from, between the stretched version and the unstretched version. And if you think about something that stretches in our body, the urinary bladder is actually a really good example. As it fills, the walls of that, which have epithelial tissue in it, have to stretch. And so this type of tissue is very, very important within our body. This one is kind of hard because in order to see it, you have to look at two pictures. So the top one here would be like empty. You can see we've got, looks like stratified squamous, but then as it stretches and it gets to be full, it still looks like stratified squamous, but notice how the appearance changes. This top part is the stretched part, and the bottom one is also rearranged. It's not as thick as it used to be. Now, keep in mind that epithelial tissue also includes glands, and so we do want to spend a little bit of time talking about glands. Glandular epithelia contains gland cells, and we have two different types of glands within the body. We have the first type, which is we're going to spend a little bit of time with, are the exocrine glands. These secrete exocrine type secretions. In other words, the secretions that they create are released onto the surface of the epithelium. In addition, when they are released, they first go through a duct or a tube to get to that surface. So the types of secretions that are typically released via an exocrine gland are the enzymes in the digestive tract and sweat. Now we need to contrast this with the other type of gland, which are the endocrine glands. These secrete endocrine secretions, which are released onto the surrounding tissue fluid as well as into the blood. So first of all, one big difference is where they are released. Okay, this goes into the tissue fluid and the blood. The exocrine secretions go onto the surface of the epithelium. The second difference is the endocrine glands are ductless, so they don't go through a little tube, whereas the exocrine glands do. We will be talking about the endocrine glands very much in depth when we get to their system, the endocrine system. Major example of endocrine secretions are hormones. Now, let's go back to those exocrine glands here for a second. We can classify them in two ways. The first type are the unicellular ones, which obviously mean that they have one cell, and examples would be either mucus cells or goblet cells. Goblet cells look just like what they sound like, a goblet. If you think of like a wine glass, that the place where the wine is called the goblet, and that's what the cell kind of looks like. The second type are the multicellulars. And the multicellulars are just that, they're many cells. And we can classify them further using three different mechanisms, one of which we're going to look at. So first way is by looking at the branching patterns of the ducts, because remember these actually have ducts within them, as well as the secretory portion of the gland looks different, multicellular gland to multicellular gland. The second way is the mechanism of secretion, in other words, how the exocrine gland actually releases the product that is being made. And the third is the type of secretion. Now, based on this classification, we are going to spend our time looking at the mechanism of secretion and how they can be different. That's probably the easiest one of the three to look at. So the first type is the Holocrine secretion. And with Holocrine secretions, we get exocytosis taking place, and this releases the vesicles only. An example of this would be mucus, or if you take a look at the diagram, it also shows you that saliva is released in this way. The black dots are the secretory vesicles, and they migrate to the surface and release their products via exocytosis. Holocrine, the cell stays completely intact, and that will be important. The second type of secretion is apocrine secretion. In apocrine secretion, we lose both some of the cytoplasm as well as the product that the cell is producing. What happens is the vesicles actually pack, cram very tightly into one end of the cell before that portion of the cytoplasm is shed. So what we do is we actually have this pattern of packing the secretory vesicles here. You can see the cell membrane pinches in, all right? That portion then pulls off of the cell. So this part of the cell is left behind. This part that's been pulled off, the membrane breaks down and the product is actually released. The cell then goes through a period of regrowth where it gets bigger, where it also generates more vesicles, and then it repeats the process. The third type of secretion is Holocrine secretion, and this is where the entire cell is packed with vesicles. So in order to actually release the product, the cell is going to burst and the cell dies. So when the cell bursts, it doesn't just empty out the vesicles. You lose DNA because you lose the nucleus. You lose all of the organelles. Nothing is left once this happens. Our sebaceous glands, our oil glands that allow our hair to actually slide through our skin, release the oil that they produce in this way. Okay, and that's what this is showing here. This one goes from bottom to top. So we get cell division down at the bottom by the stem cells, and as the cells move upwards through the layers, they produce their secretion, they get bigger, and they add a lot of vesicles. When they reach the surface, after they've reached complete maturity, the cells are bursting at that point, and they release all of the contents of the cell. The way I remember holocrine secretion is it's the whole cell that gets destroyed. So this completes our look at epithelial tissue, and in the next section we will actually start talking about connective tissue.